next speaker today, the title of his talk is Mycoplasma Hyopneumoniae Diagnostics by PCR, Tips from the School of Hard Knocks. So Albert Canturri is a veterinarian from Saragossa, Spain. He did his residency in pathology at the University of Barcelona with Kim Sagales, and he is working towards his PhD in Mycoplasma diagnostics and recently accepted a position as a pathologist at the University of Minnesota. He is a board-certified pathologist by the European College of Pathologists. So with that, I'll let Billy start, Albert. Thank you.
Thank you, Amanda, for the presentation. So today I would like to talk to you about what have we learned from hitting the wall many times. We have hit the wall quite sometimes, but we learned lots of things, and I would like to share what we learned and what others learned also from these problems. I think Dr. Bedlack did a great introduction about what mycoplasma is, so I will just go into how is mycoplasma diagnosed. So the most important part here of the idea is that unlike other bacteria, mycoplasma is very fastidious to grow in culture, and this is one of the great, let's say, problems that we have with mycoplasma diagnosis. Because for the detection of mycoplasma, we rely in the field in clinical science, right? Animals that cough. Genetic detection of genetic material by PCR, which is the most used technique, diagnostic tool that we have, and serology. The problem with that is that it's very important to remember that a detection of a microorganism is not a diagnosis. So a diagnosis needs many more things rather other than a detection, and the interpretation that clinicians or we as diagnosticians or pathologists we do is not always straightforward. So sometimes we have PCR positive results that we didn't expect, and we have to go deep and see what are they showing up, and to see if we can find the source of that positivity, etc. So and again, for to differentiate between detection and diagnosis, the context is key. So animals that cough, so the clinical picture is very important. The production parameters are also very important, and for us to have them and for you to have them also, it's very important to gather all the information possible to put it all together. Other laboratory techniques, so yesterday we talked on histopathology, so that's another good opportunity that we have to see, well, is that PCR positive result meaning something or not? So we are going to start with the hard knocks that we had. And I'm sure that you were aware of something that came up in 2018, 2019, about the detection of Mycoplasma hyopneumoniae in processing fluids. So these were two examples of those observations were published. And this is for you, how I felt, right? Or how we felt at that moment. So these were, imagine that you are in a car going, heading to a place, and you see those signs, right? Road signs, and all of them were pointing to a direction that it was, possible, and it had biological. The concept was possible, right? It made sense. So we found that Mycoplasma hyopneumoniae was detected in endemically infected herds, so that makes sense. Gill litters showed higher relative risk of being PCR positive than older parities. Makes biological sense, and knowing the infection of Mycoplasma hyopneumoniae. So let's go this way. And the detection of processing fluids in a previously negative sow farm was increased or was observed at the same time that sow farm had an outbreak. So let's go. Let's take this route and let's investigate that. Green light. So with that information, we performed a much extended study in which we enrolled. So we named it multi-farm long-term investigation of that Mycoplasma detection in processing fluids. What we did was to enroll 10 farms. 5 farms were positive to Mycoplasma, 5 farms were negative, and we followed those farms for an entire year monthly. Okay, so each month we would take 30 processing fluids, 10 from gills, 10 from P2s, and 10 for P3s and more. And we ended up gathering nearly 3,000 individual liters were tested. We pulled them and we were able to test more than 600 samples. So that was a very representative sample. And the result from the model, we did a model, and the result from the model, you will see that we saw differences in parities. So again, green light, it makes sense, right, from a biology point of view. More positivity in fall than in summer, what we see in Mycoplasma hyopneumoniae, also clinical signs on the appearance of the problems in fall and winter rather than in summer. Another green light, so let's go. And there was a marked association between the farm being positive and the detection of Mycoplasma in processing fluid. So that was another green light. So it means that when Mycoplasma is there in the cells, it is in the processing fluid. So this is maybe useful, right? What we did also was, from those, we obtained 7 DNA sequences from 3 different of the positive farms, and we investigated the did sequencing, as Dr. Rendat mentioned before. And we know that, or we found that those DNA sequences were identical between them, even if they were from different farms, and they were almost identical to the J strain of Mycoplasma hyopneumoniae. So for you to see that the J strain is a historical strain of Mycoplasma hyopneumoniae that has been used for vaccine production. So what we found out is that a lot of green lights, but we were going into the wrong way. Okay. So in this case, our hypothesis is that the origin of the detected genetic material may be associated with an environmental contamination of those processing fluids with vaccine products, right? And the hypothesis that we have is that the farm personnel that would, for example, vaccinate the weaning pigs in the morning would contaminate their hands, their coveralls, gloves, etc. And then they go to the farrowing rooms when the pigs are 2-3 days old to do the processing, and then they would contaminate the processing fluids with their hands with the vaccine product. So that was an eye-opening for us, an eye-opener that environmental contamination is a thing. And the good thing is that we were not the only ones, right? When you have a problem, it's always good to see that you are not the only one having problems about this vaccine-derived genetic material as the source of contamination. So there were 2 presentations at Leman of people observing the same thing as we did, right? So Melissa Henge and collaborators presented, they gave a talk here in 2021 in which they saw that this post-vaccination contamination was not only possible in processing fluids, but it was also possible with tracheal secretion samples. That this is the typical or the most used sample type that we use to have an antemortem detection in Mycoplasma hyopneumoniae in adult pigs. So that was very important. And she challenged us with a scenario. It was this scenario in which, imagine that we have a herd that is Mycoplasma hyopneumoniae negative, but they vaccinate. It's a vaccinated herd. So when there is a guilt introduction from the GDU, they vaccinate the guilts at the GDU, but then they may have positive PCR results on those guilts. So if you have this scenario, that's a very complicated scenario, right? Because you can have delayed shipments so that those guilts cannot enter the herd because you don't know if that's a real PCR positive result or it's just, what is that, right? You cannot take the risk. Additional testing, you might even euthanize that those animals to see if that was what was that production impacts. So that is problematic. And then Rachel Waymeyer, which was at that time a med student here, and now is a PhD student, she did collaborators in 2020. She investigated more about DNA contamination of the environment and how DNA is present in the environment and is very resistant to degradation. So what they did was intentionally spraying or the position of vaccine products in different areas of a sow farm in field conditions. And they found that Mycoplasma DNA could be detected by PCR even in the last time point of their observation, which was 28 days, even after power washing. So this gives us the idea that let's be careful about Mycoplasma DNA and Mycoplasma vaccinations and the environmental contamination of that, because we can have problems, right? And when we have Mycoplasma there, DNA, it's very hard so syngenic DNA is very resistant to degradation in the environment, and this is followed by a PCR, which is very, very sensitive. We can detect even traces of genetic material that can be positive, and this is a risk of cross-contamination within samples and environmental contamination of samples. So this is an example of pre-analytical error in testing. So this is, a very basic explanation of what we do when labs test samples. And for example, I would use the example of PCR in this case. So laboratory testing in general is divided in 3 phases. The pre-analytical phase, which is from when we collect the sample until it reaches the lab, okay, all this. And I will talk about this more later. The analytical phase, which is, for example, in a PCR, from the moment that we do the extraction until the PCR is running, we can have errors there. For example, if the internal positive control is not positive, or we have a negative control that gets contaminated, this is an analytical error. And post-analytical, which means how do we report those results? Imagine that we have 10 PCRs, and number 9, and the results of number 9 and number 8, they are mixed. This is a post-analytical error. So in the pre-analytical phase, as I said, is everything that occurs prior to the analysis, and this is where it's most important for us, or for the field veterinarians to be, it's the responsibility of the veterinarians in the field, right? So it goes from sample collection. How do we collect the samples for PCR, for example? And I will talk especially about this in the following slide, but also identification. So if we sample 30 sows, this is very typical, ear tag, changes in ear tags, or misplacement of tubes, etc. Handling, if we drop a tube and it opens, and then we see around and nobody has seen that, and we put the lid again, we have all that, right? So this is a pre-analytical error. Transport is very important. Imagine that you send samples, as Alisa said, frozen or in a refrigerated condition, and then they get stuck in at 100 degrees somewhere. So this is a pre-analytical error too. So in these errors, the sample analysis, the PCR is adopted. The problem is that maybe the sample was not the best or the most representative sample for the problem that we have. And in sample collection, and I will talk about this, cross-contamination of samples is maybe the most important pre-analytical error that we have. Okay? Cross-contamination of samples means that we are taking 2 samples and we are not doing a good job keeping one separated from the other from this surface, genetic material, contamination of surfaces, etc. And I think minimizing this risk of cross-contamination is what is crucial for us pathologists and diagnosticians to give you a good interpretation of the PCR results. So how do we collect samples for Mycoplasma hyopneumoniae? You see, this is what we use for Mycoplasma hyopneumoniae when we do a deep tracheal catheter. So we use sampling materials, a mouth gag, a laryngoscope, the snare, scissors to cut the catheter into a tube, etc. Right? So these materials can carry DNA from Mycoplasma. Imagine, the most typical example is when we take out this tube from the trachea, it can touch everywhere here, so contaminate those materials. And there can be, imagine that we are sampling 30 sows. If the first one is positive and we are not doing anything to clean those materials, there may be a DNA carryover for the other sows, and then, this is sample cross-contamination, as easily as that. So when we do this, we clean and disinfect, and when people do this in the field, we clean and disinfect the materials between sample picks, sometimes, I guess, not always. And we normally use a wipe, and we spray water, and we rinse that with water before picks. The problem is that we have been doing this, we didn't know if this worked or not, right? Nobody, we didn't have any evidence, or we don't have any evidence saying, if I do this, the DNA is removed and the risk is over. So we did a small study that was presented by Cassandra Snow in the students' session to compare various disinfecting compounds in their ability to clear or to reduce the Mycoplasma hyopneumoniae contamination of sampling materials. So I'm going to go fast for this study because you have the presentation of Cassandra if you want to have a look at her presentation. I think it's recorded. But what we did is simple. So we contaminated the sampling materials. You see here stainless steel scissors, mouth gag, and a blade of the laryngoscope. And we call that artificial or natural contamination. So in the artificial contamination, we used this a culture, Mycoplasma hyopneumoniae culture, in which we diluted that culture, and we sprayed with this nasal atomizer very fine droplets of that culture into those sampling materials, and we let them dry, and then we sample them as I will show you later. In the natural contamination, we went to a nursery that we previously exposed those animals to Mycoplasma hyopneumoniae 232 intratracheally, so we did an experimental infection. And then we used different types of sampling materials, different sets, and we cleaned them prior to disinfecting and post-disinfecting with these disinfecting wipes. So we used commercial wipes. The active ingredient is quaternary ammonium. We prepared an alcohol-based wipe, a bleach-based wipe, and then we used 2 commercial brands of hydrogen peroxide-based wipes, and our control was a wipe moistened with PBS. So the materials were sampled, as I said, prior to and post-disinfection, and not only so, what we did here was to contaminate them, to wipe them with PBS to have the cleaning sample. We used these wipes, the cleaning wipes, to disinfect them. We rinsed them with water, so that's, I think it's important, because we wanted also to have this mechanical effect of rinsing with water, that physical removal of the genetic material, and then we took a sample with a PBS one. So what we found is with the artificial contamination, so with the materials that we contaminated with the culture, these are the pre-disinfection, so this is after spraying the Mycoplasma and before applying the wipe. We see that the materials were pretty much contaminated with Mycoplasma hyopneumoniae genetic material. All the CT values were around 25. And after disinfecting with those wipes, you see that in all of them there was a reduction of the amount of genetic material, so a higher PCR CT value. But the only treatment that was able to reach a complete decontamination was bleach. Okay? The bleach-based wipe that we prepared in the lab, based in 10% bleach. Similar things with the natural contamination. In this case, we started with CT values that were higher, okay? But and all of them were infected. But again, the only one that, again, there was an increase in the CT values, but the only treatment that was able to reach complete decontamination was bleach. And in this case, one of the 2 hydrogen peroxide-based wipes. And you see that maybe it was because this was the highest one at the beginning, so there was not much DNA there to be cleaned. So take-home messages from this small study is that wiping and rinsing the sample materials decreased contamination with Mycoplasma hyopneumoniae. So, do that with whatever wipe. But if you use a bleach-based wipe, you will achieve, or we saw that in this study at least, achieved complete DNA contamination of the sampling materials. And this could be incorporated to a protocol, of good practices when collecting tracted secretion samples for Mycoplasma hyopneumoniae. Again, with the objective to reduce cross-contamination of samples. So some of the hard knocks, and then the title said tips. Some of the tips are for a complete, a correct interpretation of the PCR result. We need to reduce that risk of pre-analytical error, right? So we can do very simple things in the farm to be able to do that. So when we're taking the samples, for example, we have a card or a surface that we'll put there. If we could be able to differentiate a dirty and a clean site or zones, that would be great. As I said, clean and disinfect the sampling materials. Change gloves often. First, use gloves, and then if you use gloves, please change them often. And then this is maybe a personal thing, but every time I go to a farm, I see, I see that a lot of those catheters are not really rubber catheters, they are artificial insemination rods. And sometimes I see those bags that are already open and dust inside, and I think that's a risk. I don't like to see that. So I know, the advantages of AEI rods versus sterile rubber catheters, but I think in scenarios that have a high risk, I think it would be great to have the better sampling materials. Okay, so now we have seen, and I think I convinced you that DNA is very resistant to degradation in the environment. But what happens in a live pig, right? In an animal, inside an animal, is that DNA also very resistant? So this has been less studied, but there are some studies out there that have reported this PCR detection of inactivated, for example, in this case was RNA from Bluetone virus inactivated vaccine. So they vaccinated those, in this case was sheep and cattle, and they saw that they could detect that inert or inactivated RNA in the blood and in the spleen of those animals even 9 days after vaccination. If we have the example of humans, there is a condition or a disease in humans called cystic fibrosis. So in cystic fibrosis, this is a condition of the respiratory tract, of the airways, in which the cilia do not work very well. So there is accumulation of pus or mucus, so there is stasis there of that component. So everything that is there, the bacteria that are in that environment are not able to be cleared by the system. And free DNA, which is a highly fragmented nucleic acid from those decomposing bacterial cells or even human cells, can be identified by PCR here. So we are detecting something, DNA of a Pseudomonas, for example, aeruginosa is mainly the main one, that was there one moment, but we are just detecting remnants and of that inert remnants of those bacteria. But with a positive PCR result, we are not able to say, well, this is just dead cells or it's just an active infection. So our idea was, for example, in pigs, it can happen the same thing after we give antibiotic treatment to pigs. Maybe the antibiotic treatment is efficient, but then if we take a DNA deep tracheal catheter, if we have a PCR positive result, it may not be an active infection. It may just be the remnants of the dead cells that are there that have not been cleared by the system, right? So with that in mind, our idea was to see or to be able to answer if and for how long the DNA from non-viable mycoplasma can be detectable by PCR in the trachea of a pig, right? That is unknown, and this is something that we wanted to explore. So we did also another study characterizing the persistence of this inactivated mycoplasma, so just the DNA, the remnants of inactivated mycoplasma in the respiratory tract of pigs. And this study it's simple, so we have 8 pigs and 8 pigs, so we had 16 pigs, we divided them in 2 groups. One of them received an active Mycoplasma hyopneumoniae homogenate inoculation, right, intratracheal. And another one received an inactivated inoculum. So when I say inactivated, it means that we took that inoculum, we autoclaved that in order to kill all the bacteria or the mycoplasma, but it was still PCR positive. So these are the CT values of the 2 active and inactivated inocula, so around 29 both of them. And we gave that to pigs, okay? So, and then we followed those pigs at day 0, we inoculated them, and then we followed them very shortly after that because we thought, well, maybe, let's focus on those first hours. So we did a necropsy and we took deep tracheal catheters or tracheal secretions at 6 hours, 12 hours, and then 1, 2, 3 days, 5 days, 10 days, and 14 days. And in all of these time points, all of the animals that were alive were sampled, and one of them, we did 1 pig per group at each time point in which we did a necropsy because we thought, well, maybe the deep tracheal catheter is deep, it goes deep into the respiratory system, but maybe not so as deep as the lung or the lymph nodes, etc., right? So these are the samples that we took, tracheal secretions in the live pigs, to our antemortem sample. And then postmortem samples, we took a bunch of things, so bronchial, so deeper samples, bronchial secretion samples from a swab, bronchial or lavage fluid, and tracheal scraping. And then we thought, from a pathogenesis point of view, we thought maybe macrophages go there, and then they eat the mycoplasma DNA and then recirculate to the lymph nodes and the spleen. So maybe they are there. This is why we don't detect them in the system. So we took thoracic lymph nodes and spleen. And these are the results that we saw. So we see here in this table, it's divided in two. In the upper part is the pigs that received the inactivated inoculum. Again, dead mycoplasma, it's just DNA of mycoplasma. And we see that we did not, and this is tracheal secretions, okay, in the alive pigs. So see that in each time point, one of the pigs was not sampled because it was euthanized. And we see that there was no detection of Mycoplasma hyopneumoniae in any of the time points. And contrarily, in the pigs that received the active inoculum, so the typical normal live Mycoplasma, we see that in the first 6, 12 hours, 1 day, we do not see Mycoplasma there, but we start to detect Mycoplasma 2 days, 3 days, and the mean CT value goes down and down, meaning active infection, right? So that's the first result. We did not detect Mycoplasma hyopneumoniae in that sample. But we said, well, let's go deeper, right? We have other samples, so let's go to the lung, let's go to the lymph nodes. So we took, as I said, bronchial secretions and these other sample types, and we see that the inactivated inoculum was not detected in any of these sample types at any time point, and the spleen is not represented here because the spleen, none of the two groups, there was no detection in the spleen. And in the pigs that received the active inoculum, they started being positive at around 2 to 3 days, okay? Nothing, surprisingly, nothing before 2 days or 1 day, but then after those 2 days. So these are maybe the results or the compiled results of this study, what I wanted to share with you. So first, inactivated Mycoplasma was not detected by PCR in any of the time points. And the detection of Mycoplasma in the alive pigs was, in the first 2 days, was not dependent on the activity or the viability of the inoculum. So if you ask me where was Mycoplasma, Mycoplasma was there because the pigs got infected. But on those 2 days, we were unable to find where it was located. And we have observed this in other studies. So it's interesting. I don't know the significance of this observation, but at least it's interesting. Limitations of this study. I think this study had limitations because, of course, it was done, the first exploratory analysis of this. The CT value of the inocula was moderately high. 29 is a high CT value. I am wondering what would happen with a CT of 22 or 21 or 19. And when we have infected pigs in the peak of infection, we have this CT value. So we might look at this. And then the most important part is that those pigs were completely healthy. So the mucociliary apparatus was healthy, the immune system was healthy, but this is not what happens in the field, right? So we know in farms there is dust, there is ammonia, there is PRRS. That PRRS is known to reduce the capability of macrophages to phagocytize bacteria. We have Mycoplasma hyopneumoniae, which is the most important bacterium that decreases the ability of the mucociliary clearance to clear disease. So I think further research is needed in order to see that in field conditions. So take-home messages, last slide, Amanda. The interpretation of Mycoplasma hyopneumoniae PCR results can be complicated when we have unexpected results. This can be challenging, and I think one needs to do what we try to do, or what we did is to go deep to investigate that and to go to the source of the genetic material. The stability of DNA molecules in the environment and in surfaces is very high. We already know that, but I think it can be a risk for sample cross-contamination and environmental contamination. So we need to pay a lot of attention in the sample obtention part of the analysis. Implementing sampling protocols, maybe including bleach-based wipes, could be helpful. And also the detection of genetic material remnants from a viable PCR in the respiratory tract of pigs. I think it's still conceptually possible. So we know from humans and from that we are doing a PCR, we are detecting non-viable cells along with viable cells. But I think this needs to be further investigated in field conditions. And with that, I would like to give a big thank you to the Mycoplasma Research Lab, especially Cassandra Snow, which is the student that conducted that experiment, and the funding agencies of some of these studies. Thank you.
Thank you, Albert. That was a nice summary of routes for potential contamination of sample types that we should keep in mind. Any questions for Albert? We've got time for one question from the audience.
Thank you, Albert. That's a very great presentation. One question I have for the bleach decontamination is that have you in your study did you remove the bleach from like before you go to the downstream? Because as for ASF DNA, we also know that bleach is a very useful contaminant, but it has an effect on the downstream extraction and the PCR result. So have you included that removal of bleach from your downstream studies?
Yeah, that's a very good question, and not only because of, so the answer is yes. So after wiping the materials, we would spray water and to rinse that. So bleach and the other components of the other wipes were removed from those surfaces. But that's a good point, because one of the questions that Cassandra had was, maybe using bleach is good to remove DNA, but maybe it's too good, right? Maybe you are removing the DNA of the other pigs that maybe are positive after that. So that's something that we need to explore. But yes, rinsing with water it's in our protocol, and I think needs to be done to remove, soap, or sometimes using the other wipes, you can produce foam, for example. I think that this needs to be removed by water.
So removing bleach is before is included before you take in the sample.
Yes. Yeah. Yeah, it's spraying with water and then taking the wipe to see if there is DNA contamination. Yeah.
But then you dilute some kind of DNA if you spray with water.
Yeah, we try to. Yes, this is what I said. It's not just the effect of the wipes, but we are also accounting for an effect of the physical removal of that with the water.
Okay.
Thank you.
Thank you, Albert.